Dear students, today we are going to start with the third unit of our syllabus, cell structure and its functions. Okay, so in this unit we are going to see three major areas. So the areas are the cell, the unit of life, the biomolecules, and cell division and cell cycles. Okay, and now we are going to start with the cell unit of life. So what is cell and how can we define the term cell? So as we all know, the atoms are the basis of chemistry. Like that, the cell is the basis of biology. Okay, so all the living things, all the living organisms, all the living entities in the world is made up of the basic units called cell. So how can we define the term cell? Cell is the simplest unit of life. The first term you should remember, cells are the simplest unit of life. There are so many types of unicellular organisms, so we can define the second definition over here. They can be independent existence. Okay, they can be independent existence, and they are the simplest form. They can be alive. Okay, that is the major point. So what do you mean by alive? So yes, a protein molecule or a nucleic acid molecule or a ribosome cannot be called as a living molecule. Okay, so what do you mean by alive? They can be perform functions. Cell can perform the body's function, and it can be exist as independent small structure, and this is a basic unit of the living organism. Okay, so what is cell? Cells are the simplest unit of life. They can be exist independently, and they are the simplest unit. They can be called as alive or living molecule or living structure of any living structure or organism. Okay, so before we are entering into the detailed topics of the basic unit of life, you should know something about the cell theory. And now we are going to start with the cell theory. And before we are entering into the cell theory, you should know something about the history behind the idea of cell. Okay, so there are two names you have to remember: Anton von Leeuwenhoek and Robert Brown. So Anton von Leeuwenhoek, who was impressed from the astro studies, okay, and he has used the microscopes to observe the samples. Okay, to observe the water samples and various samples, and he has discovered the presence of microscopic living things. Okay, that is the point you have to remember. Microscopic. So he has discovered the microscopic living things, and he has discovered there are so many microscopic structures also available in the nature, available in the world. Okay. And the second major contribution is from Robert Brown, who has discovered the structure called nucleus. What is nucleus? We are going to see the details later. Nucleus is a kind of organelle present in eukaryotic cells. Okay, and these are the two major contributions you have to remember in case of the history behind the cell. Anton von Leeuwenhoek, who has discovered the biological microscopy, and who has first observed the microscopic living cells under the various samples. Okay, and Robert Brown, who has discovered the nucleus from the eukaryotic cells. And now we are going to start with the cell theory. And what is cell theory and what is the need for cell theory? The cell theory, it is going to explain the cell theory, it is going to explain the major important factors of the cell for the living world. Okay, and who has discovered the cell theory? Theodor Schwann and Sliden. So these are the two scientists in 1839. They have observed so many different types of tissue samples from both plant tissues and animal tissues. Plant and animal tissues under the microscope. And they have discovered the same as in the Anton von Leeuwenhoek. All the tissues, whatever may be the type of tissue, it may be an animal tissue or it may be a plant tissue, all the tissues are made up of the smallest unit called cell. All the tissues are made up of the units called cells. Okay, that is the major point, that is the very first point you have to remember in case of the cell theory. So what is the major point over here in case of cell theory? All the all the living organisms are made up of. Okay, that is the word they have coined. All the living organisms are made up of cells or products of cells. At this point is not going to complete the particular cell theory. The second major contribution is from another scientist called Rudolf Virchow. Okay, in 1855, and he has discovered another major point in case of cell theory, the formation of cells by means of cell division. So what is the point over here? You have to remember from the Rudolf point of view, cell division. Okay, the cells are formed from the pre-existing cells. That is the second major point you have to remember. Cells formed from pre-existing cells. So with this point we are going to conclude the cell theory. There are 2 points you have to remember in case of cell theory. The first point, all the living organisms, all the living entities are made up of cells or its byproducts. And the second point you have to remember in case of cell theory, cells formed from the pre-existing cells by means of cell division. By means of cell division. And these are the 2 points you have to remember in case of the cell theory. Okay, and now we are going to enter into the topic called the basic unit of life. Cell as a basic unit of life. There are so many different types of cells available in the nature. So how can we classify the cells into different types? Based on organelles, there are so many different types of cells and the major classes of prokaryotes and eukaryotes. On what basis we are classifying the two major categories? On presence or absence of true nucleus. So we can say pro plus caryon. So pro refers to primary or before. Okay, caryon refers to nucleus. So that means primary nucleus or before nucleus or the structure present before nucleus. That means there are no true nucleus. Okay, there are no true nucleus or exactly nuclear membrane. Okay, there are no nuclear membrane structure present in case of prokaryotic cells. That is the point you have to remember. Okay, so on the basis of the presence or absence of nuclear membrane, we are dividing the entire cell world into two different classes, prokaryotic cells and eukaryotic cells. And we are going to see the detailed internal structures of each and every type of cells. Now we are going to differentiate the two classes. What are the basic differences between the prokaryotes and eukaryotes? The first major difference. What are the differences between the prokaryote and eukaryote? Okay, the first difference starts with the size of the particular cell. Okay, as we all know, the prokaryotes are always the smallest structures present in the world. They will be around 0.3 micrometer to 1 micrometer in size. And normally the eukaryotic cells are larger than the prokaryotic cells. It will be around at least 3 to 5 micrometer to 100 up to 100 micrometer in size. Okay, and as we told in the previous classes, you have to remember the presence or absence of organelles. So what are organelles? Any structures that is being covered with a membrane. Okay, so in case of prokaryotic cells, there are no organelles present. You have to remember this point. There are no membrane bound structures or enveloped structures present in case of prokaryotic cells. There are so many membrane bound structures present in case of eukaryotic cells. So what are the different types of organelles we all know? Endoplasmic reticulum, nucleus, Golgi apparatus, lysosomes. Okay, there are so many different types of organelles. So what are organelles? Organelles are membrane covered structure. There are no true nucleus. So as I told you in the previous point, there is no true nucleus. The structure present over here is nucleoid. So as we all know, in case of prokaryotic cells, normally the DNA is in the form of the circular form, covalently closed circular DNA. It is in a supercoiled nature. Okay, That structure, the supercoiled structure is called nucleoid. There is no proteins involved in case of the DNA packing. We are going to see the details later. The point you have to remember over here, there is no nuclear membrane over here. The DNA material or the genetic material is present all over the cytoplasm as a scattered supercoiled structure, that is called the nucleoid. In case of eukaryotic cells, there is a structure called nucleus. The next point you have to remember over here, the ribosomes. In case of prokaryotic cells, the ribosomal structures are called 70S ribosomes. The S refers to Swedberg unit or sedimentation coefficient. We are going to see the details of the S. So, in case of eukaryotic cells, it will be 80S ribosomes. So, you have to remember this point, ribosomes are not organelles. Why we are saying that? Because ribosomes are just macromolecular structures. It is not being covered with the membranous structures. Okay, so that we have to call this as just a macromolecules. You are not supposed to call this as organelles. So many times we are expecting this question in a NEET exam. Ribosomes will be confused with the organelles. Organelles like nucleus, Golgi apparatus, lysosomes and endoplasmic reticulum. They are being covered with the membranes, and it is not being covered with the membrane. Ribosomes are present in both the prokaryotic cells and eukaryotic cells. Okay, the point you have to remember, ribosomes are not being covered with the membrane. So that you have to call this structure as just a macromolecule, and you are not supposed to call this as a organelle. Okay, so the difference between the prokaryotic and eukaryotes over here, the prokaryotes they will have the 70S ribosomes. In case of eukaryotes they will have the 80S ribosomes. The next major difference over here, the cell wall. Almost all the prokaryotic cells, they will possess the structure called the cell wall. All the cell wall is made up of normally the peptidoglycan layer. Peptidoglycan or the murin layers. Okay, we are going to see the structural details later, NAM and NAC structures. The point you have to remember over here, it is made up of normally the bacterial cell wall. Normally the prokaryotic cell wall is made up of peptidoglycan. Normally, in case of eukaryotic cells, the cell wall is may or may not be present. Plant cells, they are possessing the cell wall. Animal cells, they will not have the particular structures called cell wall. If the cell wall is present, normally it is made up of cellulose or the related carbon structures like pectin, mannan. Okay, there are so many different types of carbon structures over here. The point you have to remember over here, there are no peptidoglycan structures in case of eukaryotic cells. Only the cellulose or the related components will form the particular cell wall in case of the eukaryotic cells cell wall. Okay, the next point, mesosomes. So in case of prokaryotic cells, there are some structures called mesosomes or the invagination of the cell's membrane into the cell's interior. Okay, so these mesosomes gonna involve in so many functions of the prokaryotic cells. These structures, the mesosomes are absent from the eukaryotic cells. Prokaryotic cells, they are possessing the structures called mesosomes, and there are no mesosomes in case of eukaryotic cells. Prokaryotic cells, there are no centrioles, and there are centrioles in case of eukaryotic cells. So these are the major points you have to remember. There are so many other differences in between the prokaryotic and the eukaryotic cells. For example, in case of the flagella. Okay, in case of prokaryotic cell, normally the motile prokaryotic cells, motile bacteria, they are possessing the flagella. Those flagella is normally structurally simpler than the eukaryotic cells flagella. Okay, so in case of eukaryotic cells, they are possessing the microfilament arrangement of 9+2 arrangement. There is no such arrangement happens in case of the prokaryotic cells flagella, and we are going to see the detailed structural details of each and every type of cells and each and every component of each and every type of cells. The point you have to remember over here, the major points, the major differences between these two categories. The first point, prokaryotic cells are normally the smaller than the eukaryotic cells. There are no organelles present in case of prokaryotic cells, but there are so many types of organelles or membrane bound structures in case of eukaryotic cells. There are no true nucleus over here. Nucleoid is present in case of prokaryotes and nucleus is present in case of eukaryotes. Ribosome structure over here is called 70 years. In case of eukaryotes it is 80 years. Cell wall is made up of peptidoglycan or murin layers or muraminic acid. Over here in case of eukaryotic cell wall, it is made up of cellulose or its related carbon sugars. Mesosomes are present in case of prokaryotes. There are no mesosomes over here in case of eukaryotes. Centrioles are absent and centrioles are present. Flagella is arranged in a microfilaments. There is a definite structure 9+2 arrangement like that in case of eukaryotic cells. There is no such arrangement in case of prokaryotic cells. Okay, and now we are going to see the details of the structures of prokaryotic cells and what are the structures present in a external structures and internal structures and what are their role, how they are arranged in a prokaryotic cells.